Okay, now I want to talk finally about the plants for the lab practical review. I don't have the models on this. I think I have them somewhere on my phone, but I don't have a way to get that to the computer at the moment. But basically, they hopefully you've drawn your lab manual. It's all the parts that you'd be responsible for, as well as the functions of those parts. Like xylem carries the water and phloem carries the food, and things like that. Epidermis protects, for the most part. Ground tissue is makes up the rest of the body. It's where the photosynthesis would happen in leaves, things like that. Okay, so I'm just going to go through our slides for the most part for this. So the first slide we have is the pine pollen grain. So pinus is a gymnosperm, a conifer, and these are the pollen grains from the conifer. You can see, some of you might not have drawn this in your lab books, so if you are listening to this, can you please do that? And if not, I'll make it another announcement in class. But they look sort of like a Mickey Mouse to me, or a water molecule, actually. And so they have this body part. It has 2 sperm inside each pollen. So each pollen contains cells. The cells are the gametophyte that makes gametes. And so they're going to make sperm in this pollen structure. And then that sperm, there'll be 2 of them. So that double fertilization can happen later. And then you'll see these little projections here. Those we call wings. So structure fitting function, right? So they have wings here, so you can think about why pollen and wings would be useful for pollen, especially in a pine tree, a gymnosperm without a flower. And then also, why is this important evolutionarily in the sense of pollen itself? So the step of pollen, which went along with seeds and pollen, why both of those had big advances or gave it advantages to plants to move onto other parts of land. So where would they be able to move, and why did pollen allow them to do that? Then we have all the rest are angiosperms. So lilium is the lily, and this is the anther. So take a look at a picture of the anthers, the whole flower in your book. You should know all the parts of the flower: sepal, stamen, anther, carpel, that kind of stuff. But you can see here, this is a section through an anther. So we looked from, this is looking down from the top with it cut in across the middle, or kind of horizontally, I guess, sliced. So here's the filament, that's the stalk, and then this would be the anther, part here. And then you see we have these pollen sacs inside the anther, which is the male part of the reproductive system. And they make pollen. That's what all these cells are inside. Or pollen grains, which contain the pollen. The pollen grain contains the sperm. So in the anther is where they make the sperm. Then we have the ovary. So you see it looks different. The ovary, this is when if you were taking and cutting, you'd be cutting that bulb part at the bottom of the flower, ovary part. And if you look down on it from above, this is what it would look like. And basically the ovules, you can see here, are forming in the ovary. Those are going to be fertilized potentially by the 2 sperm from the pollen grain. And then they would turn into a seed, and the seed would get dispersed. So you can think of it too, like the ovary is all sort of together, whereas the anthers have these kind of lobes to them. Anthers look more like antlers to me than the ovary does. The ovary is very much like an O shaped. So that's flowers, but don't forget to also look at the flower parts, the flower model, because you are needing to know those parts. And I think I put on your packet the parts to know. Excuse me. If not, you wrote them down during class, and if you didn't write them down, try to get a contact with the classmate. So the leaves, monocot, dicot, which is which? Oh, same with flowers. So the monocots flowers are going to be in groups of threes, so 3, 6, 9. Whereas the eudicots flower or dicot are going to be in groups of 4 or 5. So if I were to bring in a flower and ask you if it was, a monocot or a dicot, you would be able to answer that. Leaves, you can also tell from their sections, monocot, dicot. So there's two main ways. One is looking at the ground tissue. So all this stuff is called mesophyll, mesophyll. And there's two types of mesophyll in eudicots and only one type in monocots. So in eudicots, and this would be that model that you saw, there's this top layer of these kind of elongated cells we call the palisade layer, palisade mesophyll. And then down here, the kind of more spread out, is the spongy mesophyll. You can also say mesophyll. So in dicots, you have the two layers, which it's not why it's named that, but that makes sense. The di and the dicot is two layers. However, it has, and in the section you'll probably only see one major vein, and that's the difference that you can tell monocots, because monocots have many veins, all parallel, right? So if you were to look at the whole leaf of a zea mays, that's corn, so a monocot, you would see those parallel veins, which is what you're observing from this section. Eudicots, if you looked at those leaves, you'd see branched veins. So parallel veins going to indicate monocot, and these are all obviously not round, so that's how you know it's a leaf, not a stem or a root. And if it has two layers here, you know it's a eudicot. And then also the model for the leaves as well. Oh, I wanted to mention, I'll just go here, oops, that on the model for the leaf, there was two labels backwards. So the cuticle is the very outside layer of the leaf, the surface coating on the top and the bottom, actually. And then the first layer of cells on the top and the bottom of the leaf is the epithelium, or the epidermis, maybe we should say. I'll say epidermis. The epidermis is the first layer of cells. So basically, if we're looking here, the cuticle will be outside of this, and here's the epidermis. That's the first layer. So cuticle is like almost the red part, and then the first layer of cells is the epidermis. And then this is mesophyll. Here you can see stomata open up to the air, let carbon dioxide out and oxygen, or oxygen out and carbon dioxide in. With stems, you need to be able to tell it's a stem, not a root. And then if it's a monocot or a eudicot. So here you notice that the eudicot has all of its vasculature, the vascular bundles around the outside in a ring, while the monocot has its vascular bundles all scattered throughout. And we decided that these look like little alien heads in the slide. So those are the monocots. If you were at lab when we talked about that. And I'm not going to go through everything here. You can look at the labels to figure that out. That's different than roots. I'll come back to apical meristem in a minute. So these are the stems. These are the roots. So stems have a bunch of vascular bundles, And so does the monocot root, really. But they're still sort of contained in the middle. So we really don't have anything smack dab in the middle in stems. But we do in roots. Both of them are in the middle. Now, very, very center, very tight. All the vasculature is all in this one bundle, and it's an X of xylem, and the green's phloem. That's a eudicot. So when it's all concentrated in the middle there. When the vasculature is in a ring, but it's still a middle ring. So you see it's not like scattered everywhere, when it's not a ring on the edge like the stem was. It's a monocot. So monocots have the ring of vasculature in their roots, not just one vascular cylinder. You should also be able to recognize whole specimens. So basically, a taproot versus a fibrous root, and which one has which. And then how you would tell if it's a taproot or a fibrous, how you looked at that and figured that out. And then root hairs, that's the projections here. They're actually an extension of the cell, what the purpose of root hairs are. And then there's a symbiotic relationship where the root hairs associate with a fungus. So know what that term is. We just talked about that one in the fungus section too. And I should also I'll mention at the root, there's also a symbiotic relationship, not just with the fungus and the root hairs, but also with the root and nodules. So those little bulbs or little growths, they're kind of round, spherical. Those are nodules. They grow on the roots. And we saw them back in the chapter about bacteria, because it's really the bacteria growing inside those nodules. And they're going to help with nitrogen, getting nitrogen to the plant in a usable form. So that's the bacteria, the rhizobium bacteria. And then the mycelium is going to be the symbiotic relationship of fungus and root hairs. And that is for water, like getting more water and getting more minerals like phosphate, phosphorus. By increasing surface area. And then you'll need, of course, the slide models for all of the above. And yeah. Secondary growth, so you definitely want to go through that. So to know, like, okay, the very innermost is going to be what? And then, so, the cork cambium, straw bunch of circles. So the cork cambium is going to be here, let's say. And outside the cork cambium, so that's where the stem cells are in the stem. So the cork cambium. Then outside, every time it grows, it's going to put down phloem. Phloem's to the outside, right? The phloem's the food. So phloem gets put down to the outside of that vascular cambium. To the inside of the vascular cambium is xylem. So you're putting phloem to the outside and xylem to the inside. So the first layer goes like this, like our vascular cambium. Okay, so we got our phloem inside and our xylem inside, our phloem outside. And then what happens is basically there's another cork cambium out here, and it's going to put down the cork, which is on the outside, And the phloem, the cork cambium, and the cork are all the bark. So what happens with the vascular cambium? It still puts down more rings. By that I mean it grows next year. So our next new one will be blue. So our dark blue. So that's our middlest one is our oldest, in this case our secondary xylem. This is our primary, our oldest primary xylem, the very first one we put down. So the primary xylem is the most innerward. Then this is our secondary xylem. It's the one we just made this second year. Then we have our phloem on the outside. And the thing is, when we grow that way, our new layers are closer to the cork cambium. So we'll end up like this. So here's our cork cambium. You can see the primary phloem's on the far outside. That's the first phloem we made. It's dead now. And the new phloem, the secondary phloem, is close to the vascular cambium. That one's where you're going to be moving your food through. The phloem won't die really till it gets to the very end before it falls off. So really all this phloem should be functional right now. The xylem on the other end, this is definitely not working. So the xylem that's in the middle is dead xylem. It's all clogged with resins. Versus the phloem out, and then the xylem, the next to the closest to the cork cambium or to the vascular cambium is going to be the ones that are carrying water, and then the phloem, both these guys actually, will be carrying food, and then we have our cork cambium, and then the cork cells, and then the epidermis. And so all the phloem plus the cork cambium plus the cork are all the bark. And eventually the phloem falls off or gets shed, whereas the xylem stays all the time. Okay? So that's what I've got for you about plants. And then as long as you just know the gross anatomy of the plants, meaning what they look like in 3 dimensions with the models or real-life specimens. Okay, well I hope that helps, and good luck studying. I will see you tomorrow.